I am honored to be here with such an attentive, bright-eyed audience. I'm particularly pleased here to be with the seniors who are the ones wearing the blue shirts. I have a blue shirt on. I want to be a senior. Yeah, right. I'm going to talk about some work that I've been doing with one of my colleagues at the University of Notre Dame. This is very recent work. We haven't published this yet, so please don't talk about it too much outside the room. I hate to be scooped by you guys going back to your laboratories. The work we're doing is extraordinarily interdisciplinary. We work with biologists, and Rebecca's talk just fits in perfectly with what we do. We work with computer engineers. We work with physicists. I'm a chemist. And so one thing to remember as you guys are going through your education, don't compartmentalize your learning, because things interact with each other to a surprising extent. I want to talk about biological transformations, where one thing goes to another. And the example I'm going to use is a frog. How many of you have ever been in a pond and caught a frog? A couple of you, a bunch of you have. So look around. If you don't know what frogs look like, ask the people whose hands are up and they'll tell you what frogs look like. Now think of the coolest biological transformation you can imagine. What if we take this frog and we introduce this frog to a beautiful princess? And of course, if that beautiful princess kisses the frog, we will generate a handsome prince. Now, as Rebecca taught us earlier, frogs and handsome princes are both made of cells. But somehow the cells from the frog have to be transformed into the cells of the handsome princess. This is called a frog to prince transformation. I wanted to study this phenomena. I think this is the coolest thing. And I went to Dean Crawford and I said, Dean Crawford, I'd love to do this, but we need to have a colony of frogs and we need to have a colony of handsome princes. And Greg said, we can do the frogs. It's too much money. Princes, you know how much they cost to keep going? We can't do that. So we went a little bit backwards in time and development. And we're going to look at the transformations that occur as an embryo is fertilized and goes through the first stages of development. So Rebecca talked to us about that earlier. We're going to look at it from a chemist's point of view and look in details. We're looking at Xenopus lavis. It's a frog. It's the African tree claw frogs. They're frogs. You've seen them. And it goes, you start out with an embryo, an egg, and this fertilized egg goes through all of these transformations, blastulas, organs forming, all of this stuff going on as the cells evolve from one to another, ultimately end up with the frog. We're interested in the chemical transformations that occur. And now I'm going to have to say a word to you, a second thing to remember. First thing to remember is don't compartmentalize science. The second thing to remember is that you and I and frogs and handsome princes are more or less mostly protein. And you look at yourself, you look at your thumb. My thumb is made out of thumb proteins. My ear is made out of ear proteins. My nose is nose proteins, etc. My liver is liver proteins. Kidneys are kidney proteins. And we want to characterize the evolution, the transformation of the proteins from an egg into an embryo into a frog. Now, of course, you look at the embryo and you look at the frog, you look at the prince, and you say they kind of look a lot alike, actually. And Prince Charles is, at any rate, they both have noses, they both have mouths, they both have eyes. And if we were to study the transformation of frog to handsome prince, we'd be interested in the transformation in the proteins. How do they evolve? How does it happen? So here's the Xenopus embryo. It has just been fertilized. It's gone through one cell division. Now this egg, this ovum, this embryo, goes through a series of cell divisions, one after another, and these are incredibly coordinated. You could call it a dance of life, a symphony of life, whatever metaphor you want. But these are all controlled by the basic biology within this embryo. And we want to understand the details associated with it. So we're going to take a fertilized egg, and then we're going to watch what happens as it develops over the first day. And the way we do this is we take fertilized egg, and then we'll at various points in time, we'll take a single embryo, we'll take that single embryo, we'll tear it apart, and we're going to analyze it chemically. That's what my group does. That's what my students and researchers do. Before we came along, this science was done in a form of artesian science. You know, you talk about artisan bread, artisan beer, artisan football teams, artisan science. This is groups of science working in their laboratories, beavering away, studying one of these proteins in the embryo and how that protein changes as a function of time. Here's some data that just appeared in Science in August. This is real scientific data. And scientists look at these bands and look at the intensities and wave their hands and make arguments about how biology happens. We wanted to speed this stuff up. This took a group of 5 scientists 5 years to generate these bands. Within this organism, within this Xenopus, this frog, there's about 5,000 different proteins. It would take each and every one of us working for 50 years to completely characterize the evolution of proteins within this organism. That's just too long. So my group decided to do the same amount of work in 2 weeks, and the way we're doing it is using some of the facility at the University of Notre Dame. We have one of the world's best proteomics facility in the world. We have visiting scientists come by, we show off this instrumentation, this laboratory, and these scientists say, that's really cool, I'm jealous. And we use an instrument called an Orbitrap Velos, which is a really cool sounding name. It's a box, it's about the size of a dishwasher. Inside of it is vacuum plumbing and physics-like accelerators and filters and mass analyzers. We take the proteins from this organism. I forgot to mention, I have the dean here, and I don't often get the dean's ear. These instruments are really expensive. And here's a quotation for the latest instrument. We just got the quote last week. And it's for the Orbitrap Fusion Basic. It's the basic Orbitrap Fusion, which list price of $900,000. And then you throw on the bells and whistles. You got $1,027,000. We got a discount. So we only need $827,300, Greg. And we can buy the state-of-the-art instrument. So we're using not quite the state-of-the-art instrument. We also do some chemistry. I showed this slide when we practiced to Dean Crawford. Dean Crawford said, "Ugh, chemistry." And Dean Crawford's a physicist, and so of course that's what he'd say. I just want to put this out here for those of you who might be studying chemistry, that chemicals do have use. And these are isotopically labeled chemicals, so physics has use too. I'm not going to tell you how we use them. But what we did is industrial scale science. We took these embryos and we studied their proteins, and we identified 4,000 different proteins within these embryos and how their abundances, their amounts change as a function of time. We took the amount of work that would take all of the people in this room 50 years combined to accomplish, and we did it in a week, well, 2 weeks worth of time on this Orbitrap instrument. We have created a database that will be mined for decades as people go through and use the information we've generated in order to understand the details of development. We took our data and threw it into some sophisticated software, and the sophisticated software pulled out patterns of how the proteins change as a function of time. And here's a graph where we have 6 different patterns that we observe. In order to understand this, I have to teach you some biology. How many have taken biology? How many haven't taken biology? So you guys probably haven't taken biology. You've heard about DNA. How many of you can spell DNA? The central paradigm or dogma of biology is that DNA will be transcribed into RNA, a different related molecule, and this RNA in turn will be translated into protein. So the proteins that we're observing have come from RNA, which in turn came from DNA. One group of proteins we observe does a steady decrease in abundance and concentration and amount as a function of time. These proteins are the egg proteins, and they're being consumed. So if you think about the frog to handsome prince transition, here we're seeing the frog proteins disappearing. We're seeing the egg proteins disappear. There's another group of proteins which are kind of noisy, but these proteins pop up as a function of time and then slowly decay away. These are proteins which are made from RNA that came from the mother. This is cool. You have an egg. The mama puts her DNA inside of the egg, and then she also puts inside some of this RNA, some of this message, this message in a bottle. And this message in a bottle gets cracked open once the egg gets fertilized, and this RNA will be turned into proteins. But the maternal RNA will disappear with time, and so do those maternal proteins. There's another burst of proteins appear where the embryo starts doing its own thing, where the embryo's DNA is turned into RNA, is turned into proteins. In turn, these proteins affect the production of other proteins, and other proteins. At this particular stage in development, the organism is generating its own organs. So we have here these 4,000 different proteins. They're not doing the same thing. Some of the proteins are decaying, some of the proteins are popping up and disappearing, some of the proteins are coming up later. As this single-celled embryo evolves, develops into a fully grown organism. Now you might say, this is kind of cool in an intellectual way, but really, why do I care about eggs or handsome princesses? The reason we do this is deadly serious, of course, because we're interested in studying birth defects. And I'll just give you one example where Xenopus lavis, the organism we're studying, the frog we're studying, is a model system for studying congenital heart disease. And what happens, and now we'll go back to, is that these proteins are talking to each other and they're working in concert. Some of these proteins are coming up in time, some are going down, they're moving along. Think about the score for an orchestra. An orchestra is playing a symphony, beautiful music. How many of you are musicians? We must have musicians. We have 3 musicians here. Study music, kids. Here we have a score from a symphony. You'll have the flutes playing their notes. You'll have the piccolos, the oboes, the horns, the violins, the drums, all playing their own notes at specific times during the orchestra. And that's where the studying these organisms becomes important, because if those notes are not played properly, if they're played in the wrong order, then it results in birth defects, congenital diseases. Here's my research group standing outside of OSHAG. Those of you who are Notre Dame alums will recognize OSHAG. I want to highlight one person in my group. This is Liang's son. He's a postdoctoral fellow. He came to my laboratory from China, from the Dalian Institute of Chemical Physics. He has a PhD, and he's coming to my lab to spend 2 to 3 years in order to get additional training before he goes on to start his own lab. Some of the people in this picture are undergraduates. These are people who are in their 3rd and 4th year of study at Notre Dame. And they are spending anywhere from a few hours a week through a full summer in my laboratory working on these research projects. Most of the people in this picture are graduate students. After you get your PhD, after you get your bachelor's degree, you go to graduate school. In my group, it takes students 5 years. It takes an average of 4 years and 10 months of additional work after you get your college education in order to get your PhD. Then you have your PhD, you do a couple of years of postdoc, and then you become a faculty member. So the steps: you guys need to go to high school. You guys need to graduate high school. Once you've graduated high school, you go to university. Once you're done with university, you go to graduate school. Once you're done with graduate school, you do the postdoc, and then you get my job. And this sounds like a long time and a long effort, and it is. It's not quite as bleak as it sounds for the parents in the room. Once you get your bachelor's degree, the rest of it, you don't have to pay anything. So graduate students are supported on research fellowships and are supported on teaching assistantships. Postdocs are supported on research fellowships. I also need to point out my colleague, Paul Huber, and I need to put the halos on these guys. These are hot people. Paul is on the left. He's a colleague of mine. One of the beauties of being at Notre Dame is it's a small school, and you have to play well with each other. And Paul's office is around the corner from mine. And my postdoc and Liang's son and I were talking about this research idea, and I said, I bet you Paul, he has Xenopus. We can go talk with him. and went around the hall, talked with him, introduced Michelle Bernicke, his graduate student, she's just about to finish up, worked with Liang Liang and generated all of this data. It's a fun collaborative effort at Notre Dame, and after hearing Rebecca's talk, we'll talk some more on that. Funding, this takes money. The research group takes about $1 million a year to operate. Much of our funding comes from the National Institutes of Health, the sequester, and now the lack of funding of the federal government is not helping us. We also get funding from industry. We work with 2 pharmaceutical companies, Eli Lilly down in Indianapolis, and MedImmune, which is in Washington, D.C. It's a big biopharma company. And both of those companies are interested in using our technology to characterize the therapeutics, the drugs that they make. And then finally, of course, the University of Notre Dame provides lots of resources for us, for which I am incredibly thankful, Greg. Thank you very much.